Hi everyone, and welcome to Micro 2. We're going to get started here with Unit 1, where I'm going to talk about respiratory cultures and pathogens. If you have the Mayhem book, this is in chapter 32 of your book. And we're going to talk about respiratory infections as well. So let's get started. So in order for a respiratory tract infection to occur, organisms have to come in contact with the mucosal epithelium and begin to multiply. So if someone comes up to you and coughs on you and you breathe it in and the organism sticks to the mucosal epithelial cells, that's how we end up getting sick. Organisms may be colonizing normal flora or invading pathogens. So you can be a carrier of strep A even if you're colonized, and they may or may not be pathogens for you. Colonization basically means organisms multiply and grow under optimal conditions without actually causing harm to the host. A great example of colonization is when you have a catheterized urine specimen, and those organisms tend to live around the urinary catheter, but they don't actually cause harm to the host. That's an example of colonization. So there are several factors that lead to a respiratory disease, including adherence factors, toxin production, normal tissue function that is impaired, and the host defenses that are invaded. Adherence factors, this would refer to organisms that can attach to the mucosal surfaces because they have pili or fimbriae, which are short pili. In toxin production, certain organisms like diphtheria and pertussis produce diphtheria and pertussis toxins. Normal tissue function is impaired due to bacterial infection. Organ support or tissue cushioning can be affected. And in the host defenses that are invaded, bacteria are not phagocytized due to their capsules, such as strep pneumo and Haemophilus influenza type B. So the respiratory tract can be divided into the upper respiratory tract or the lower respiratory tract. The upper respiratory tract includes such organs as the nose, the throat, the oropharynx, and the nasopharynx. In the lower respiratory tract, that would include the larynx, the trachea, bronchi, bronchioles, and alveolar air sacs of the lungs. So there's a number of ways that the respiratory tract is protected from infection, and that would include things like nasal hair. Nasal hair actually filters out and traps organisms when we breathe those organisms in. Our respiratory tract includes lysozyme. Lysozyme is a chemical that kills the cell walls of gram-positive organisms. We have antibodies. These antibodies recognize and destroy foreign invaders. Think about secretory IgA and how that antibody is included in mucosal surfaces. We have ciliated epithelial cells. These ciliated epithelial cells trap more organisms. We have macrophages and cellular defense mechanisms, including phagocytes. We have coughing, sneezing, and swallowing. Those three actions expel bacteria and irritants. And then we have normal respiratory flora. This normal respiratory flora competes with pathogens for nutrients. So there's a number of types of samples that we can collect to help diagnose upper respiratory tract infections. Some of those sample types would include throat swabs. We're especially looking for Streptococcus pyogenes, or we could be looking for Streptococcus pyogenes and all other pathogens. We can also collect mini-tip nasopharyngeal swabs where we're looking for things like Neisseria meningitidis, or we're screening for such things as MRSA. And then we can also see if sinus, eye, and middle ear aspirates are collected. Now, these may be treated as respiratory specimens, but more likely they are considered a wound culture where we will look for anaerobes. Let's talk a little bit about throat and nasopharyngeal cultures and kind of what we're looking for when examining these upper respiratory specimens. First thing first, we don't want to do gram stains, or gram stains are not necessarily performed unless we're looking for something very specific like diphtheria or yeast. Gram stains are not really going to help us because there's so much normal flora. That's the same reason why we don't do gram stains on stool cultures. The media that we're going to set up for throat and nasopharyngeal cultures would be an anaerobic blood agar or an aerobic blood agar with agar cuts, especially if we're looking for strep A. And we would also want to set up a bacitracin and SXT disc. That would help us to look through many plates very quickly in order to screen for Streptococcus pyogenes. If we wanted to do an entire throat culture for all pathogens, we would set up 2 blood augers, One would be anaerobic and one would be in CO2. That's going to pick up your strep pneumos. We'd also have a chocolate auger in CO2. That's going to pick up your Haemophilus influenza. And then we're going to set up a MAC and maybe a modified Thayer-Martin, depending on whether or not we're looking for gonorrhea. McConkey auger is, of course, going to pick up all those enteric and non-fermenter pathogens. So like stool cultures, there is a number of organisms that are considered normal flora of the upper respiratory tract, and there's a number of organisms that are considered pathogens of the upper respiratory tract. Some organisms that are normal flora would be things like alpha and gamma strep. That would not include Streptococcus pneumoniae, okay? Neisseria species, diphtheroids or Corynebacterium species, Micrococcus, anaerobes, Strep B, and then maybe a couple colonies of enterics, non-fermenters, and yeast, as long as that is in conjunction with a lot of normal flora. Those things can be normal. Now, things that are pathogens were going to be group A strep. Group C and G strep can cause sore throats, but they don't lead to glomerulonephritis or rheumatic fever like strep A does. Another pathogen of the upper respiratory tract would be Corynebacterium diphtheriae. That causes diphtheria. You can also have gonococcus, which is Neisseria gonorrhea in the upper respiratory tract or in the throat. Arcanobacterium, that's that gram-positive rod that causes a lot of symptoms just like strep A. And then if you have heavy growth of strep pneumo, Haemophilus influenza, Moraxella catarrhalis, yeast, Staph aureus, enterics, and non-fermenters, those would be considered pathogens if you had a lot of growth of those compared to the amount of normal flora. Now, nasopharyngeal cultures are pretty specific, and a lot of times those are collected to screen for MRSA and also to screen for Neisseria meningitidis. Here we're looking for carriers of MRSA and Neisseria meningitidis. Remember, 30% of the population does carry Neisseria meningitidis in the nasopharyngeal area. Use of a nasopharyngeal culture might be most useful for diagnosing things like Bordetella pertussis, Corynebacterium diphtheriae, mycoplasmosis, and respiratory viral infections such as influenza A or B or parainfluenza or RSV. Let's go ahead and start to outline Haemophilus species. This is a new organism for you to learn. So a Haemophilus species is what is considered a tiny gram-negative rod. They actually look like Coccobacillus, and some textbooks say they are tiny gram-negative Coccobacillus. They are pleomorphic in rod shape, but they can actually look like Coccobacillus as well. There's some nice images in your textbook if you have your textbook. They are catalase and oxidase positive. They will reduce nitrates to nitrites. They are fastidious and capnophilic, so they do require chocolate agar usually, or horse blood agar, and they do require a 3 to 10% CO2 atmosphere. They're not going to survive refrigeration, so most laboratories do not refrigerate respiratory specimens. Okay. Haemophilus does require blood factors for growth on lab media, and some species are totally normal, while other species are pathogens. 10% of humans have Haemophilus species as normal upper respiratory flora. Haemophilus influenza may or may not be a pathogen, but usually we do identify it and let the doctor decide. Like I said, Haemophilus species does require either X, V, or both factors in order to get it to grow. Factor X is also known as hemin, and hemin comes from hemoglobin. This is a heat-stable salt from hemoglobin, and it is heat-stable. Factor V is also known as nicotinamide adenine dinucleotide, or NAD. This is an important molecule in many metabolic reactions. It is concentrated in intact red cells but unavailable to the organism unless the RBCs is lysed, and it is heat liable. There's a number of possible pathogenic species of Haemophilus, namely Haemophilus influenza. That's probably our most important Haemophilus pathogen. There's also Haemophilus parainfluenza. This is an opportunistic pathogen that causes endocarditis, bronchitis, and inner ear infections. We have Haemophilus ophrofilus, but the new name for this organism is Aggregatibacter. And then we have Haemophilus ducreyi, which can cause genital tract infections. Typically, Typically, the non-pathogenic Haemophilus would include Haemophilus hemolyticus and Haemophilus parahemolyticus. So Haemophilus influenza is what we call a tiny gram-negative rod. Some textbooks call it a tiny gram-negative coccobacillus. It may be normal upper respiratory flora. If it is found in the lower respiratory tract, though, it is considered a pathogen. It may actually be a pathogen of the upper respiratory tract as well. It just depends on how much other normal flora is present. Haemophilus influenza does require both the X and V factors for growth. Chocolate is the primary isolation media. On chocolate agar, both X and V factors are available because the organism will look tannish green or possibly mucoid and may smell mousy. It does have both X and V factors available because the red cells in the media have been lysed. Haemophilus influenza is not going to grow on sheep's blood unless there's a source of the V factor. Now, Staph aureus can provide the V factor, and so Haemophilus influenza on blood, if there's Staph aureus present, will satellite around that colony of Staph aureus. It will, however, grow on rabbit and horse blood, but just not on sheep's blood without the presence of Staph aureus. So here's a nice picture of satellitism. Staph aureus provides the V factor. The X factor is already in the media, and then colonies will satellite around Staphylococcus aureus and appear like little gray dots. The media here in this picture is sheep's blood agar. That provides the X factor, and Staphylococcus aureus provides the V factor. Other pathogenic Haemophilus species and their requirements would be Haemophilus parainfluenza, which does require the V factor. It's a common cause of endocarditis, and it can occasionally cause a respiratory disease. Aggregator Afrophilus used to be a Haemophilus species, and it does require the X factor. This can cause blood infections, endocarditis, and brain abscesses. And then finally we have Haemophilus ducreyi, which may actually coexist with syphilis, and syphilis, of course, is caused by Treponema pallidum. Haemophilus ducreyi causes what's known as a chancroid. This is a sore on the genitalia that kind of resembles a syphilis chancre, but it has a more ragged edge. Our non-pathogenic Haemophilus would be Haemophilus hemolyticus and Haemophilus parahemolyticus. Haemophilus hemolyticus is normal flora of the throat and upper respiratory tract, and it does require both the X and V factors. It's beta hemolytic on rabbit and horse and human blood, and it may resemble strep A. On some medias like horse and human blood, that's where it's going to resemble group A strep. It's a big reason why we use sheep's blood instead of horse or human blood, because this particular organism will not grow on sheep's blood. Haemophilus parahemolyticus does require the V factor. It is beta hemolytic on horse blood. One of the best ways to differentiate Haemophilus species from one another is by using the X and V factor tests, and also to see if the organism will create beta hemolysis on horse blood. So in this picture's example, we've got Haemophilus influenza that's been made into a lawn on the plate. We've placed the X factor, the V factor, and the XV factor on the plate, and you can see that Haemophilus influenza satellites all the way around the X and V factor disc and between the X and V disc, and that says that this organism does require both X and V. There's a few other ways to identify Haemophilus influenza in the laboratory. One such way is by using fluorescent antibody tests, where here we use antigens of Haemophilus influenza, and if they're present, they're going to attach to antibodies tagged with a fluorescent dye. Other tests we can do are slide agglutination tests, also known as latex particle agglutination tests. Sometimes these are performed in the emergency room to rapidly diagnose Haemophilus influenza meningitis. They can do these latex particle agglutination tests on fluids such as cerebrospinal fluid. Haemophilus influenza can cause all different types of infections. Type B, Haemophilus influenza type B, is the most pathogenic strain of Haemophilus influenza, leading to the vaccine development. There's about 16,000 cases of children that are infected with Haemophilus influenza as a respiratory infection a year. But Haemophilus influenza can cause a number of different infections, including pharyngitis and laryngotracheobronchitis, epiglottitis, which is swelling of the epiglottis, sinusitis, eye and ear infections, bronchitis and pneumonia that are often secondary to a viral infection. Haemophilus influenza can also cause endocarditis, cellulitis, and meningitis. And there's a vaccine that's been developed to reduce Haemophilus influenza meningitis cases. The vaccine is made from toxoids or fractionated material. There's a couple of ways to treat an infection of Haemophilus influenza. One way to treat it would be by the use of ampicillin. Ampicillin would be used for non-life-threatening cases, or cefotaxime or ceftriaxone for life-threatening cases. A second choice would be tetracycline. This organism should always be beta-lactamase tested. We want to make sure that it is sensitive to penicillin, ampicillin, and cephalosporins. So on this chart here, I've tried to summarize which organisms need which factors and what they will do on horse blood agar, whether they're going to demonstrate beta hemolysis or not. So if you'll notice, probably the most important one is Haemophilus influenza. It does require both the X and V factors, and it will not hemolyze horse blood agar. To set up the X and V factor test, what we're going to do is we're going to get a nutrient broth of some sort. Usually that's BHI broth, and we're going to put one colony of organism in that BHI broth. We're going to take that BHI broth and we're going to make a line across a BHI plate. Aseptically, we're going to place the XV and the XV factors on the plate, and then we're going to look for growth around those factors at 24 hours once they've been incubated capnophilically. We're going to see which factors the organism needs for growth, and because of that reason, we can determine which organism is which based on which factors have been used by the organism. So there's a couple of different discs on the market for X and V factors. In the upper left-hand side, they are rectangular discs. These are called taxo discs, and you can kind of see the pattern of Haemophilus influenza. And then on the bottom right, we have the same discs, only they're smaller and round, and you place those significantly apart. And then what you will see is that the organism grows around the X and V disc. It would probably grow between the X and V disc, except for the fact that they aren't close enough together for that to happen. So let's talk a little bit about upper respiratory tract infection signs and symptoms. People that have an upper respiratory tract infection generally present with a headache, rhinitis, which is basically a runny nose, red, swollen throat and uvula. They might have a gray coating on the tongue and enlarged lymph nodes. The types of specimens for culture for upper respiratory tract infections would be nasopharyngeal swab, sinus washings, or pharynx or throat swabs. Usually the organisms that we're looking for that are pathogens would be things like Haemophilus influenza, Streptococcus group A, or Corynebacterium diphtheriae. The signs and symptoms of a lower respiratory tract infection would include things like a cough, and sometimes you're coughing up bloody and purulent sputum, chest pain, and dyspnea, which is difficulty breathing. Specimens for culture would be sputums, bronchoscopy secretions, or transtracheal aspirates or lung biopsies. Bacterial species associated with these types of infections would include things as Streptococcus pneumoniae, Staphylococcus aureus, pretty much any enteric, Moraxella catarrhalis, or Legionella species. So that is it for Unit 1. If you have any questions, please reach out to me via email at tbailey@southeast.edu, or you can call me at 402-437-2761. Thanks and have a great day.